Hi guys, so my name is Pearl Bang,
I'm Alexander.
And I'm Micah.
and we did a project on Synodesmus dimorphus as a biofuel and bioremediation tool. So some algae benefits are that it's easy to grow, all it needs is sunlight, water, its nutrients, nitrate and phosphate, and carbon dioxide. Algae is also sustainable and a friendly approach, so it's eco-friendly and easy to use. It's also a bioremediation of wastewater tool. So the Central Valley is known for its high nitrate phosphate content, so algae would be perfect to cure this wastewater.
So some of the methods that we used to study Pseudomonas dimorphus involved a hemocytometer, which was used to do cell count to determine its growth over an 8-day period. At the same time, we also ran spec data on the algae at 750 and 680. At 680 nanometers, we were measuring pigmentation, and then at 750 nanometers, we were measuring the optical density of the algae. We also performed gravimetric analysis to get a mass yield of our algae, and then we as well did a lipid extraction to get a biomass yield.
So we first constructed a bioreactor, which combined algae in an optimal culture medium, which contained the nitrate and phosphates. We then counted our cells using a hemocytometer, which uses a microscope to count individual cells. And we coupled that with a spectrophotometer so that we could get, like Alex was saying, an optical density. From those, we derived a growth curve so that we could graph the exponential growth and eventual plateau of the culture. Then we took a gravimetric analysis of S. dimorphus from culture, which gave us a wet weight, a dry weight, and then a combusted ash-free dry weight. And then at the end of this 8-day period, which is about halfway through the 17-day experiment, we analyzed the entire biomass and calculated lipid content by extracting lipid and dividing by the total biomass. We conducted this experiment in parallel between the BG11 culture medium and a more relevant pistachio wastewater culture medium to get an idea as to how readily deployable S. dimorphus would be in the field.
So in conclusion, we did these are graphs of our BG11 absorption at 680 and 750, and we also did a relevant graph of pistachio water absorbance between 680 and 750. And we also did the nitrate and phosphate content in our pistachio water. And as you can see, after day 3, there's no more nitrate or phosphate in the content. So alcoholism is really important. So that's really important for us to know, like, what are the factors that contribute to that. So our goal is to use Drosophila as a model to study alcoholism. There are alterations in gene expression in the brain when alcohol is consumed, and there are neuroadaptations that contribute to alcoholism. And this is really important because some symptoms and effects of alcoholism include memory loss and irregular heartbreaks, and it could lead to stroke and unhealthy habits in the future.
So the reason that we chose to use Drosophila is because it's a model system. It's very simple to work with. It has a short life cycle. It allows for a large population test. It's easy to do genetic manipulation on. And then the fruit fly's internal alcohol concentration is very similar to that of humans at each stage. So for our experiment, we have 3 fly groups. We have a negative control where they do not consume any ethanol, an experimental where they do consume ethanol, and then a positive control where we have no ethanol consumption but an antisense RNA against mRNA target. And then we will be measuring about 15 to 20 flies, and we will be running we will be running 4 trials.
And so, as Alex mentioned, we're using an antisense siRNA, which is mimicking the effect of that microRNA expression that we expect to be following alcohol addiction in the fruit flies. And so the net effect of the siRNA exposure or the in vivo microRNA exposure is a net repression of protein expression. And the way that we can measure that is by isolating and purifying proteins and then characterizing their presence through a Western blot. And potentially, if we wanted to further study the enzymatic qualities of the proteins, maybe we expect them to accrue some conformational abnormality or other problem, we could conduct a high throughput enzymatics in a 96-well microplate, as we have listed here. And so altogether, this provides a very useful, economical, and manageable model to study human neuropathologies